Hi, my name is Flynn, and today I want to introduce a new deep learning framework for enhancer discovery. The title of my presentation today is called Where Before What, a weakly supervised framework for precise enhancer localization. Through the phrase where before what, I hope to convey an intuition on what is a weakly supervised method and how we can utilize that for enhancer localization. So enhancers are a type of regulatory element that increases the transcription likelihood of a particular gene. Therefore, it is very important to map out cell type specific enhancers to find genetic drivers for various diseases like cancer. Earliest methods for enhancer discovery have focused on unsupervised approaches because they lack training data. For example, Chrome HMM is a hidden Markov model that discovers different states in the chromatin. However, because it is an unsupervised method, the discover chromatin states doesn't really mean anything. So human researchers would have to go and assign the type of regulatory element to each chromatin state. With the advent of massively parallel reporter assays, it is now possible to identify transcriptional enhancers directly based on their functional activity and use that as label training data for supervised methods. One of those assays is called StarSeq, and the rough experimental procedure is as follows. Given a target genome, you fractionate the DNA into 500 base pair fragments, and each fragment is inserted in front of a luciferase gene as a part of a plasmid vector. Each vector is transfected into a target cell. And if the fragment does contain an enhancer, then it would increase the transcription of the luciferase gene, which could be detected by mRNA gene expression. However, there are a few shortcomings to this experiment. First, it has a low transfection efficiency, meaning that it has a high cost on running this experiment on new cell lines. Second, it has a low resolution. The peaks that were called from StarSeq are about 500 base pairs in resolution. However, the smallest enhancers are about 50 base pairs, and the largest enhancers can go about 1,500 base pairs. Therefore, StarSeq cannot evaluate the precise location of small enhancers. Lastly, StarSeq can only evaluate the fragments outside of its native epigenetic context. The assay can only evaluate the ability of a sequence to increase transcription. And from previous studies, we know that the epigenetic context also contributes to the identity of an enhancer. Therefore, it's also important to take that into consideration as well. With all of these shortcomings in mind, we want to build a deep learning model that amends these problems. We want to train a model that can transfer its predictions to new cell lines to lower the cost of enhancer discovery. We want to also somehow increase the resolution. And lastly, we want to incorporate epigenetic context as input to our model as a part of the decision-making process. For cell types that we have StarSeq experiments on, we downloaded the corresponding epigenetic information from the ENCODE data portal. For open chromatin, we downloaded the signal tracks for ATAC-seq and DNA-seq. We also downloaded the signal track of ChIP-seq for these 4 types of active histone marks. Our central hypothesis is that the interaction between open chromatin and active histone marks provide a platform for TF binding and serves as a basis for enhancer activity. Here is an overview of our workflow. Our training data consists of 4 KB genomic windows with positive examples containing StarSeq peaks and negative examples sampled from random background regions. For each 4 KB window, we extract the signals from these epigenetic profiles for each 10 BP bin and organize the values into a matrix. Then that matrix is fed through our neural network to output a binary prediction of whether it contains a StarSeq peak or not. If the 4 KB region does contain an enhancer, then we utilize a procedure called GradCam to backpropagate the gradient to find regions that were important in making a positive prediction. Those regions are a subset of the 4 KB windows and define our core enhancer region. In addition, we can use the same GradCam procedure to produce feature justifications as well. I will go into further detail on gradient-weighted class activation mapping as a weakly supervised learning method. Intuitively, If I gave an image to a human to classify whether there is a dog in this image, and the human says yes, implicit in that answer is that the human has already found where the dog is. The human would say no if it did not find the dog. Therefore, implicit in the classification task is actually a localization task. And that is particularly what GradCam does. GradCam extracts the localization underneath the classification. It was first developed in the computer vision community to debug their models and enhance interpretability. For example, to classify whether if there are bikes in a set of images, because bikes frequently co-occur with humans, the model might predict the presence of bikes with humans as a feature. These decisions can all be visualized using GradCam. And so the details of GradCam is as follows. The convolutional filters in a neural network learn to uh, select features that are important in making an accurate prediction. If I input an image through a convolutional filter, it produces an activation map that highlights the relevant features important for an accurate prediction. For example, if I input an image of a dog, one convolutional filter might output an activation map highlighting the nose of the dog. Another convolutional filter may highlight the eyes of the dog, and many different convolutional filters highlight many different features. A linear combination of these activation maps produces a heat map localization of where the object is. And that is particularly the power of this weakly supervised method, is that we can infer the location of objects in our image without exposing that information to our model during training. We can utilize the same idea in the precise localization of enhancers as well. For a 4KB window, we can use GradCam to find regions, subset of 4KB windows that are important in making a positive prediction. And those subset of regions are what we called core enhancer regions, or object detected localization of enhancers in this window. In the following sections, we hope to evaluate the quality of our positive predictions as well as our core enhancer regions. Here, we show training statistics for our DECODE framework. Overall, we see that given the epigenetic features as input, we can accurately classify whether there is an enhancer in the 4KB window. Further, we performed two types of internal validation. On the left, we left one cell line out and trained on all other cell lines. And on the right, we left one chromosome out and trained on all other chromosomes. For both types of internal validation, we achieved high metrics showing that we can learn to predict enhancers accurately based on epigenetic features. Here, we show example predictions from our DECODE model. For the positive regions, there is a lot of activity in the epigenetic profiles. In particular, there's single peaks in the DNAs and double peaks in the active histone marks that is characteristic for enhancers. In contrast, we barely see any activity across all signal tracks for the negative predictions. We benchmarked our trained DECODE model with a state-of-the-art enhancer prediction model matched filter on the ENCODE Enhancer Challenge dataset. Matched filter relies on shape matching filters to extract features for classification with linear SVMs. It was the winner of the ENCODE Enhancer Challenge. For experimentally validated transgenic mouse enhancers, our model outperformed matched filter for all tissues, some with about 15 to 20% margin. We could do so because number one, we used a large scale functional assay as training data, and number two, we used convolutional filters that are better at modeling the interaction between epigenetic features. For GradCam, we could backpropagate gradients to lower levels of the network designed specifically to output an important score for each feature at each location, also known as the feature-wise score. Or we could backpropagate gradients to higher levels of the network to output an important score for the aggregated features at each location, also known as the pointwise score. Further, we could use the latter score to refine our positive predictions to find the core/refined enhancer regions. Here is a case study of feature justification and prediction refinement on neural progenitor cells. We can see that there are two enhancers in this 4KB window. In the feature-wise score, DNase H3K27AC and H3K4ME3 are all emphasized, and that importance is passed to the higher levels of the neural network as reflected by the emphasis on the same locations in the position-wise score. Then we can set a manual cutoff to define the refined region as regions above a certain threshold. For genome-wide sliding window inference on neural progenitor cells, which does not have StarSeq experiment data on yet, we found 17,622 positive predictions and 23,505 refined positive elements after second pass with GradCam. The refined positive elements are only around 13% in coverage compared to the positive 4KB windows, yet accounts for a higher proportion of TSS sites, indicating a strong transcriptional impact from the GradCam refinement. It is also shown for important regulatory regions like enhancers, they exhibit negative selection pressure, as any mutation in these regions would likely cause dysfunction that prevent these traits from being passed on. Therefore, enhancers should be more conserved, and conservation could be used as a metric for evaluating the quality of enhancers. Here, we quantified interspecies conservation using the 100-way phylogenetic FASTCon score, showing that our original 4KB positive windows are more conserved than genomic baseline, with even more conservation in the refined regions. We quantified intraspecies conservation with the enrichment of rare derived allele frequency SNPs. And for these two sets of SNPs from Nomad and Peacock, our original predictions are enriched for these SNPs, but refined regions are even more enriched, corroborating with our conclusions from FASTCons. Lastly, we performed disease causal variant mapping on the original versus refined predictions using stratified LDSC. We see that for enhancer regions found in neural progenitor cells, those regions are enriched mostly only for neurodevelopmental and psychiatric phenotypes. Further, refined predictions has higher and more specific LDSC enrichment compared to original predictions. The increase in statistical power could be attributed to our compact annotations. The ability for GradCam to shave off irrelevant regions in the object detection step greatly increases the statistical power for causal variant mapping. And lastly, I want to say thank you. A special thanks to my PI, Mark Gerstein, my mentor, Jing Zhang, other co-authors, Jason Liu, Donghong Li, and other members of the Gerstein Lab who have helped me with this project. Thank you.